So we will continue with cloning vectors. We were discussing PBR322. Now this is a constructed vector which is constructed from the plasmid, E. coli plasmid. So this combine the feature of certain other plasmid and whatever is required to be used as an ideal plasmid, that is how it is constructed. Okay, and this is used as multiple purpose cloning vector. So this is the first constructed vector of size 4361 base pair, that is about 4.4 kilo base pair you can say. So it can easily you can insert about around 5 more than 5 6 7 base pair of kilo base pair of DNA as compared to you can say if you see the original plasmid, which can only take up the less insert size. So the transformation efficiency is inversely proportional to size of vector. So less is the size, more good is the vector. Okay. Now this is developed by Bolivar and Rodriguez. So the nomenclature depend on these two scientists who have uh, discovered this vector and constructed this vector. Um, P stand for plasmid, B for Bolivar, R for Rodriguez. 322 is a specific number which is assigned to this vector, and this is how it is different from rest of the vectors. So unique identity is given to the vector. Now if you look at the features, it has origin of replication which is from Colivan, you can say plasmid. Then it has got two selectable marker, ampicillin resistance and tetracycline resistance as a selectable marker. Then it has 40 restriction endonuclease nuclease sites. 11 of them lies in tetracycline gene, and I think around 6 they are present in the ampicillin resistance gene. So we will see how these are useful restriction endonucleoside when they are specifically present within the antibiotic resistance region. So what is the advantage? Because of the small size, easy to handle, then it has got two selectable marker. Later on we will see how they are very important and they impart good or easy selection. Then amplified to 1000 to 3000 copies. So size is small, usually 100 copies are formed within the cell, okay, of plasmid, but this can form 1000 to 3000 copies because its size is small. Okay, so next is the cosmid. So cosmid are constructed by using cos site from lambda phage and plasmid, certain features of plasmid. So let's see how these features are combined. Um, you know what is the significance of cos site? A cosmid is a type of hybrid plasmid that contain lambda phage cos sequences. The cos DNA circularizes phage genome. Therefore, in cosmid, it impart in vitro packaging. Then cosmid contain origin of replication from coal even, and it contain ampicillin resistance gene and multiple cloning site. So these are the features which are imparted from plasmid. So advantage is that they have cloning capacity up to 45 kilo base pair, and they are especially used in the construction of genomic libraries. Okay, so next is the phasmid. It is a plasmid that contain origin of replication from F1 phage, and it can be replicated as a plasmid and also be packaged as a single stranded DNA in viral particles. Now we'll be discussing yeast artificial chromosome, that is known as YACS. So yeast is used Saccharomyces cerevisiae, vector that enable artificial chromosome to be created and cloned into yeast. So this is known as yeast artificial chromosome. This is engineered DNA molecule used to clone the DNA sequence in yeast. Okay. So yeast is a eukaryote. So certain eukaryotic genes are also cloned and studied in yeast cell. Okay. Even the human genome project was started in yaks. Okay. So let's see what are the features. It has telomeric sequences from yeast. It has centromere sequences from yeast. It has origin of replication from yeast and E. coli both. Then it has selectable markers from both yeast and E. coli. Then it has restriction endonucleosides such as EcoR1, BAM-H1 and many others. Okay. Now advantage is that this is used for the cloning of DNA fragment larger than 100 kb to 3000 kb and very useful in physical mapping to analyze very long DNA sequence. Okay. So in the mapping what we are doing, we just try to identify the sequence of gene located on the chromosome. To map the entire human genome, which is of the size 3 into 10 raised to 8 base pair, it requires more than 100,000 plasmid clones. If we are using the plasmid, then we have to use so many plasmid. But if yaks are used, only 10,000 yaks are required to clone this. So you have to handle a very less number as compared to say plasmid. So the yaks are required in less number because they take up the large size of the insert. Disadvantage is that it is very fragile and unstable and loss of entire yak during the mitotic growth. So handling is not very easy. Lot of loss of experiments are there when we are doing the experiment with yak. Lot of failures are there. Now if you look at the journal structure of vector, if you have gone through all the vectors, so if I made one vector which contain all the features of all the vector, okay, just to give you the idea that what exactly is the vector. So the vector should have origin of replication, which is very important. Otherwise, if they don't have their own origin of replication, they cannot multiply within the host cell. Okay. Second is antibiotic resistance gene, which is used as a marker. It may be 1, 2, 3. Okay. So if they have 2, it offers the advantage in this selection, good selection. And these antibiotic resistance gene, they have the target site for restriction endonuclease so that they can be cut and you can insert the foreign DNA within them. Then there are certain vector with LAGZ gene. Okay. Now all these features are not present in all the vectors. Okay. Different vectors are there and they are with different features. So some of them also have LAGZ gene and this consists of multiple cloning site. So this impart again a good selection method. So they are all artificially done in a piece of DNA to construct the vector. So nowadays we are using all constructed engineered vectors. Okay. The technique used for the gene transfer. So we'll discuss in the next video. So this completes here the vector.